Hi, today I'm talking about phenotypes and test ordering in VariantGrid. As an example, I'm going to use one of our old cases written up as a clinical report. So I'm going to copy the phenotypes from here and paste it into the software. So what's happening now is the server's trying to fuzzy match the text to ontology terms. So HPO, Mondo, OMIM, etc., and the aliases. So we have our results, and you can see the terms have been highlighted according to what was matched. So orange is for HPO and light green is for Mondo. You can hover the mouse over the terms to see the details. So the process isn't perfect, and you can see there's some pretty important information left out. The thinning of the corpus callosum wasn't recognized. So in this case, we can use the autocomplete to try and get the correct terms in there. And we'll do this and we'll add it on to the end and then match it to an HPO term. The earlier you get the phenotypes into the system, the better. So ideally, you'd start at the test ordering phase. We'll fill out a pathology test order now. So the columns with the percentage show the percent 20x coverage in our capture kits. As well as our tests, we can also bring in the panel app panels. So these don't always agree, and what we're going to do here is just add these two genes as custom genes to add on to our standard pathology test order. So we'll send this order through. And because the medical system loves paper, you can generate a PDF. Here we have an overview to see how we link everything together. So now we'll look at what kind of information is stored against a gene. This is the gene page. There's a lot of info here, but we're going to look at the gene disease and gene phenotype links. So gene disease links come from the Gene Curation Coalition, or GenCC database, which collects gene disease evidence links from many different sources. Next is PanelUp, the Oz and the UK versions, which I'm sure you're familiar with, and they're a way of creating gene lists and sharing them. And next are the terms associated with the gene via the different ontologies, so Mondo, OMIM, etc. There are a lot more of these, and the level of certainty is not always clear. You can go to the ontology page in our system to see when it was retrieved and what else it links to, so any classifications or patients with that term. Now we're going to use this patient phenotypes to filter VCFs. So this is a variant grid analysis. The top node represents de novo variants in a trio, and the other nodes below it are filters. We can click on a node to see the results, as well as adjust the filters in real time. So to start with, I'm opening a mode of inheritance node. This filters to those GenCC curated genes and optionally the zygosity for that patient's diseases. So again, this information comes from GenCC, and you can see it on the gene page. The next node is a gene list node. You can filter to a pathology test, a gene list. You can type in some custom genes, but at the moment, we're going to use some panel app panels. You can autocomplete off the panel name, and we'll then call the API, take a snapshot of the latest version, and store the retrieval date. For the final node, we're using all the ontology terms associated with the patient. So this is quite broad and returns a lot of results, so you may need some further filtering or curation. Okay, that's it. Thanks for listening.